This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWIV, This Week in Virology, episode 968, recorded on December 23rd, 2022. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Fort Lee, New Jersey, Dixon Despommier.
Hello, gang. Out the window, it's gotten gray and very cold. That's all I have to say.
This morning when I came here, it was rather warm. It was raining.
Yes.
It's gotten colder. It's 1 degree C, and we're expecting flurries. Also joining us from Western Massachusetts, Alan Dove.
Good to be here. It is still 49 Fahrenheit, 9 Celsius here. I'm expecting that to plummet very soon. It is blowing about a full gale, gusting up into the 40 to 50 mile an hour range, and the skies are just stacked up castles of clouds blasting through at high speed. It's pretty dramatic.
So poetic today, Dr. Dove.
We must say that the National Weather Service, and you asked me not to talk about the weather, but they had never had a winter alert like they did last week, ever. It affected more people in the United States than any other winter alert ever.
Okay. But nothing happened of it, did it?
Well, yes, indeed. People died.
Thousands
people
of flights canceled, and a lot of people lost power.
Well, here nothing happened, and this is all that matters. Okay, let's move on. Also joining us from upstate New York, Brienne Barker.
Hi, great to be here. You may notice that it is snowing outside here, and it is 20 degrees Fahrenheit.
Right.
And from, see, I grouped all these similar weather places together. And now we have from Austin, Texas, Rich Condon.
Well, get this. 31 degrees.
Right.
We're about a day ahead of you guys. It was 16 last night.
Wow.
See that?
Wow.
16 Fahrenheit. 31 today. It's not going above freezing today. And for the next, let me see. Tonight, the forecast is 20, then 23, 28, 33. So we got 4 days of this.
Kiss all that citrus
It is going to
fruit.
go above freezing during the day, but
Kiss the citrus fruit goodbye.
yeah, well, you know.
You have quite the shining sun on your wall there, man. That's quite.
Oh, yeah, that's Tinkerbell. And there's not much I can do about Tinkerbell because unless I
No, don't worry about it. It'll move in the course of this 6-hour show.
Moves around.
Yeah, it's Rich Henge.
This week, Nell Zeldy had a Tinkerbell on his nose. I said, "You got Tinkerbell." He had no idea what I was talking about, of course, and I don't blame him. It's a TWIV joke. All right, we are winding down. This is the second of two episodes for 2022. Good riddance, 2022.
Say it second to last.
Second to last, yeah.
It is the penultimate episode of
Penultimate.
2022.
Maybe that should be the title. Penultimate viruses.
I thought 2022 was okay.
It was okay.
Yeah, 2022 wasn't too bad for me.
Better than 21. Yeah.
It was certainly an
That's
improvement
true.
over the preceding 2 years.
Oh, yeah. Oh, absolutely.
Yeah, that's true. The further we get it from those 2 years, the better. All right, we have a lot of science for you today, which is good. And before we do that, I have two announcements. First of all, as the year winds down, so does the deal at vaccinated.us. You see Brienne's wearing a Spike shirt there.
Is that like a hoodie, Brienne?
No, it's a long sleeve t-shirt.
The long sleeves are nice, yeah.
I think I might need another Spike t-shirt.
You guys all got gift certificates from me, right?
Mine just arrived yesterday, and I wore it to work out this morning. The gym is the main place that I wear graphic tees.
Did you get yours, Dixon?
I have not ordered one yet, actually.
But I sent you a gift certificate.
You did?
Yeah. Unless I
Check
thought
your email, Dixon.
that T-shirts don't fit you and I didn't send them. I don't know. It's possible.
That was sent via email?
Yeah, I sent gift cards. I'll check into it.
Thank you.
I highly recommend the long sleeve t-shirt.
It's under the heading.
I'm into long sleeve t-shirts.
Vince, it's under the heading chopped liver.
Chopped liver. Anyway, you can buy a Spike T-shirt anytime, but if you buy it in the rest of 2022, some of the profits will go to support our science shows. And the the funds we've gotten so far from vaccinated.us have not been insignificant. Okay, so keep buying.
Right.
Go to vaccinated.us. Everything's available for this promo. Someone wrote me and said, "Can I get the long sleeve?" Yeah, it's all part of the promo. Just buy whatever you
I picked up
want.
a mug in addition to the shirt. The mug has not arrived yet, but looking forward to that for my coffee.
So go to vaccinated.us, pick what you want, put it in the cart. When you go to check out, you put Microbe TV, all one word, no periods, in the promo code section, and that will tell them to send something to us. Get this. Here, 2 weeks ago, I ordered a bunch of gift certificate for all the Microbe TV hosts. I forgot to put the promo code in. What a schmuck.
No.
Can you believe it? Here's a guy who talks about it every week. So I had to email Matt, and he said, I saw that come in, and I took care of it. So they also have... Yes, sir. Go ahead.
I don't know if you can see this or not, but way in the background over here where my finger is pointing.
Yes. I see a Twiv mug. Twip mug.
And Twiv.
Good boy. The other thing is you can get gift certificates, right? If you have a company of thousands of employees and you want each of them to wear a Spike shirt, that would be cool. We get a lot of money out of that. You just go and you put their emails into a field and they all get emails and you pay for everything and they just go pick it out. That's very cool. That's what I did here for Microbe
And
TV.
they would be absolutely perfect for your volleyball team.
That's right.
Yeah.
Because
Spike shirt, yeah.
Spike shirt,
that's right.
yes.
So the other thing I thought about was that, they had a promo last year during the World Series. For every stolen base, Taco Bell would give you a free taco. If you walked in and said, somebody just stole the second base, oh, yeah, you get a free taco. In the National Football League, when they score a touchdown, they spike the football.
Yeah.
We could give away
Why
a T-shirt
don't
every time.
we give away a taco whenever someone gets vaccinated? Why don't we do something useful instead of stealing bases?
We gave away, I believe, if you took your vaccine card to Krispy Kreme, you could get a donut.
There were a
cool.
bunch of promos. I think Duncan
I
might have
didn't
done
know
a
that.
promo
Nice.
as well, and some other places did. There was
Think of
definitely a lot
something.
of that sort of
Is
thing.
the first one with a plaque assay that says "Hello, Vincent"? We will give them a Spike T-shirt. How's that? If you spell it out with the plaque.
This gives me an idea. Next year, we're gonna do one of these Twivs live, okay? Because
we'll be very sorry. We will
having
be
people
very sorry.
having people chatting is a lot of fun. We'll do one next year.
I would agree.
All right, so that's vaccinated.us. And then we have from Miguel, who wrote, I was wondering if you could post a couple more positions for us. So we had already posted some positions with the Mosquito and Vector Control Division in Harris County Public Health, somewhere in Texas. These are this division is responsible for running and maintaining a mosquito virus testing lab. Interesting. And so we will put links to these jobs descriptions. It looks like there are two, a virologist and a virology laboratory coordinator. So if you are interested in viruses and mosquitoes in Texas.
And you should be.
And that's in the Houston area.
Houston?
By the way, I had to look it up on a map, but that's Houston.
Okay. Into this, and now for the science. as Mark Crislip says, and now for the literature. This first one came out some time ago, and we've had a slew of guests, so I haven't done it yet, and it's still in preprint. But I got an email from the senior author, Jean-Michel Claverie, who I've met many times and who Rich has met in the giant virus meeting. And he wrote, this is a very funny email. So this guy's French, right? He's in Marseille. Dear Vincent, I'm amazed and flabbergasted by the incredible buzz that my very last paper triggered. There was nothing new, except I was just emptying my drawer from all these ancient viruses we never had time to characterize in detail and publish. With an altmetric attention score of 1700 plus, this preprint is among the top 0.1% of all bioRxiv preprints. Just try to search Claverie permafrost virus in Google News, and you will see that basically all media with the slightest interest in science ran a piece on it. Sure, I cannot imagine that Twiv would not devote 30 seconds of its broadcast time to mention how a non-event such as that can suddenly make the news worldwide. I just love the way he writes. So he sent the link. Proud to become finally famous after 42 years of anonymous career. Bien amicalement, Jean-Michel Claverie. So, Jean-Michel.
Well, now we've devoted at least 30 seconds to it, so
Yeah,
we're going to move on.
I was going to say I'm going to start my stopwatch.
I would say echo in Italian. Here it is. What do you say? Voilà, right? Voilà in French. So this is a preprint, an update on eukaryotic viruses revived from ancient permafrost from Jean-Michel Claverie and Chantal Abergel, his wife. They both worked together, and we did wonderful paper from her lab this year on the structure of chromatin, or the DNA in these giant viruses. Really fascinating. That will make the top papers when we do that next year. First author is Jean-Marie Alempic. Do we have multiple?
Yes, two first authors.
And Audrey Lartigue. So this is all about the permafrost, right? What is it? A quarter of the northern hemisphere is got frozen ground, permafrost, right?
Yep.
And
Well, it used to be frozen.
it used to be, but now it's
Less
thawing.
and less frozen, yes.
So as the planet warms, this is thawing. And so what's going to come out of it, right? And it talk a lot in this paper about bacteria coming out of it, and we know that there are bacteria in there. But they say this is going to not be in the final paper. The situation would be much more disastrous in the case of diseases caused by revival of an ancient unknown virus. And this is because he thinks all the bacteria that come out we could take care of with antimicrobials, right? But he said, viruses, we can't. This could be a disaster. And so that's why the press takes a hold of this, right? Because
Well, there are a bunch of reasons why this gains traction. First of all, it's very easy to get the gist of this paper. I mean, you just skim the abstract and you've got the basic, they found more viruses in permafrost.
yeah, that's right.
Second, it's got a bunch of cultural tie-ins that people are going to get immediately. You could come up with a bunch of cool headlines for this. I mean, there's the whole, it came from the Arctic expedition. You've got the slime and, all this, the blob, sorry. So there's the horror movie background of it. And then, of course, it ties into climate change, which is a big subject. And so you can link to that. So this is made for headlines. That's
and it got him, as he said. Yeah.
That's why it got picked up by everybody, because it's an easy paper to understand, and it's got
But it's funny
a
because
lot
he
of
was
important
just
stuff going on.
clearing out his drawer.
Clearing out the drawer. He had this stuff sitting around.
I
Question-
think that the way that he handles some of the sort of biosafety things in the writing is also kind
Yes,
of neat.
that's
Sort
funny.
of, both at the end where he's like, well, obviously we can't do this with viruses that infect other things, and in the beginning, in the intro where he's, here's the literature on the subject. Happily, there have been no follow-up studies attempting to revive these viruses.
So he goes through a few of the viruses that have been recovered from old materials. And of course, the first was influenza virus from permafrost since 1918. And by the way, Jeffrey Taubenberger, whose group did that, was on TWIV yesterday. So you should check that out. He talks all about it. And if you watch the video, he shows me one of the little pathology specimens that they used to carve out the genome. And he has some of the old sequence gels as well. They did 10 bases at a time. It was just painful. It took them 10 years. All right, so that's 1918. Then he talks about smallpox virus DNA in a 300-year-old Siberian mummy buried in permafrost. Did we do that paper, Rich?
I don't recall.
Yeah, neither.
We've done a few papers talking about poxvirus DNA out of old samples.
Yeah.
There was one with Vikings,
Yep.
right? And I don't know if we did the permafrost one, but the Viking guys were older than 300
Yeah.
years. So
And these samples are from various places, right? Some of them are like core samples and that kind of thing.
The ones that they did, the Claverie lab, was a core sample from
These are-
Siberian permafrost. So they previously got 2 infectious viruses, giant viruses that affect amoeba. They published
actually, it
that.
just occurs to me that they ought to stick in the final paper, they ought to stick a figure with a map that shows
Yes.
where these sites are.
That's a good idea. And
Yeah, where did you drill the holes?
The point here is that we had these 2, which we published a while ago, and you may think it's pretty rare, but actually we have a bunch in our lab. And so we have more to tell you about. And that's what this paper is. And there are a couple of interesting ones, and they have some pictures of them as well. So they got 7, right, viruses out of permafrost. 4 of them were Pandora viruses. These are very cool. They're big. The largest known genomes, linear DNA up to 2.6 million base pairs.
So I think an important feature of this is that the way they screened for these, they took samples and they processed them some so that they would have virus left over. But then they seeded them onto cultures of amoebae.
Right.
And looked for the amoebae to show some sort of cytopathic effect. So right away, you're screening for these types of viruses, giant viruses that grow on amoebae. And as they point out at the end, you're looking for viruses that actually cause some sort of cytopathic effect on the amoeba. So that's a pretty. I mean, this is. I'm not bashing it. Just so you understand that this is a fairly narrow screen, and so it comes up with these kind of creatures.
Right,
and by the way, I just wanted to back up to the sample thing because I said something incorrect. These are not primarily core samples. These are Table 1 actually lists GPS coordinates and descriptions of where these are from, and it's a bunch of different places. Looks like a few in Russia and some other.
Aren't they ice cores though?
They've surface soil. They've got a river sample, something below a lake, melting ice wedge, woolly mammoth stomach contents.
Interesting.
Fossil wolf intestinal contents.
They basically have a gram of sample that could be liquid, mud, solid soil. Right. And then they get a pellet from it.
And
these are different from the original because the original were from ice cores from Siberia, and they were dated, right? So,
and going back to Rich's point about the amoeba, you could imagine doing this with other types of cells to do a screen for other types of viruses, but then there might be potential safety concerns because who knows what those viruses are.
Yeah. Do you want to grow out a virus from permafrost that grows well in human cells?
Right.
Maybe
well,
not.
you probably should do it under BSL-4 conditions, right? I would guess if you're going to do it. But if you did it, people are going to scream and yell.
Then yes.
Right.
What is the oldest virus that they recovered? How old?
Well, here. These, right?
They got one here from
48,000?
48,000 years
Before
ago.
the present?
It's kind of when you were born, Dixon. Aren't you the 48,000-year-old man?
That's an unkind remark.
It's actually a kind remark.
I wish
you look good for your age.
Yeah.
Thank you. I feel great too. I just ran across a blurb on Yahoo News, which is my primary news source, that was from the BBC, and there was a laboratory in England, I believe, that through DNA amplification from tundra samples, they were able to reconstruct a 200 million year old ecosystem.
200 million?
2 million. But still, that's a pretty long time ago.
Right.
And the DNA has lasted that long enough to allow them to reconstruct the plants and the animals that live there.
Ah, yes, cool.
And they made some interesting observations, and I wondered if they had thought about looking at viruses that way also.
Yeah, I'm sure they could have found viral genomes, yeah.
So So you raise an interesting point, Dixon, because that's metagenomic sequencing. That's sequencing. These are live viruses that they recover.
Yes.
I hear you.
Because they put them on amoeba, and they get what as Rich said, it's killing the amoeba. So they say, okay,
modern amoeba. These are modern amoeba.
Modern amoeba. Yeah, that's a good point.
Sorry, live viruses. I don't know.
Infectious.
That's a legitimate infectious. Okay.
Exactly.
So Pandora virus. Then they got Pytho viruses. The Cedrat virus, which I hadn't heard Cedrat yet, but there you go.
These names?
These are different from Pandora's. The smaller circular DNA genomes, 400 to 600 kilobase pairs. They have a cork-like structure at their extremity. I've always liked that.
Cork-like
French, the French, they called it a cork-like structure.
Cork-like structure.
They're thinking about corks a lot in France, right? And it does kind of look like a cork, and it does come out to let the DNA out. So it's
that's true. The only other
pretty
place they think about cork a lot is, of course, Ireland.
Yes.
Thank you very much. I'm here Wednesdays. Try the veal.
Then they have a Mimi virus. The only Mimi virus rescued from permafrost is called Megavirus mammoth. It's a. I guess this came from the
Came from the mammoth, I
mammoth
think.
samples, yeah, which was frozen in the permafrost, right?
Wait,
Yes.
they got it from the stomach contents?
Yeah.
Well, isn't that plant viruses?
Could be.
And this guy.
No, this is not a plant virus. It's an amoeba.
It's an
virus.
amoeba virus, but the amoeba could have been
It could have been in the
Right.
mammoth.
There might be
stomach.
plant viruses in there.
Exactly.
So that's Mimi virus, and then Pac-Man
one.
virus. This is the good one there. Pac-Man viruses. They're acanthamoeba infecting virus, distantly related to African swine fever viruses. Only and those viruses infect pigs, right? Now, Rich, those are Asfarviridae, right?
Yeah.
Different family from Poxviridae, right?
Correct.
Are these transmitted by vectors, insect vectors, or am I thinking of something
Ticks.
else? Ticks. Okay.
The natural habitat is a wild boar in Africa, transferred by ticks from one animal to another. Doesn't do much, but if it gets into the more the wild pigs in Europe or domesticated pigs,
right?
it's lethal.
Yeah.
So it's a big problem.
Well, and you see where they got this one from?
Yeah.
It's from the frozen intestinal remains of a Siberian wolf.
right.
Yum.
It's 27,000 year old. So that's old too. Double-stranded DNA, 400,000 base pairs. Okay. Pac-Man virus. That's so. What was the original reason for Pac-Man? I guess I don't know. I forgot. Why is it called? Does anyone know why it was called Pac-Man virus? I mean,
No, but I can imagine, if it's
doesn't
got one
take
of
much
these star
imagination.
things, it's probably got a mouthy looking thing on it.
all right. I got the paper. Pac-Man virus, a new giant icosahedral virus. Let's see if I can quickly grab it. Yeah, there must be some feature which makes it look like Pac-Man. Okay, that's fine. What else? I think that's it, right? Yeah, so there are phylogenetic trees. There are some electron micrographs, and that's basically it. I don't know where this is going to end up. I suspect this is not like a nature science cell thing, right? But who knows these days? This could be a nature microbiology or a cell host microbe, right?
Yeah.
Maybe. We'll see.
If you look in figure 1F, there is a picture of the Pac-Man virus,
Oh,
and I
thank
can
you.
kind of see Pac-Man.
If in the paper that you found, Vincent, if you look at figure 1B, it looks exactly like a Pac-Man.
That's funny.
Oh, yes, I see. Yeah, it's open on one side, kind of.
Okay.
Yeah.
Well, I like the French. They are very jovial about their naming, right? Right. So there was initially Mimi, which was microbe mimic. That was cool. Then they had Mama, and then Moo Moo. They're taking this whole
Got to
thing.
keep the theme. Yeah.
But Rich, do you remember at the last Giant Virus meeting, Matthias Fischer made a song based on these?
Yes,
Mama, ooh, remember?
Yeah, it was Bohemian Rhapsody.
That's
Bohemian
Bohemian Rhapsody.
All right,
That's
yeah.
right. That's right.
He will do it again at the next Giant Virus. Actually, he promised to send me a recording, which we could play here. That would be fun. So that's the finding. And a couple of discussion points that I think Brianne actually brought up earlier. I got to get to the discussion before I can do the discussion. Okay, biohazards, right? First of all, what Rich said, they're only looking for amoebal viruses, and they're lytic. So as they say, many small non-lytic viruses escape our scrutiny, which is what no American would write, but it's I like it very much. The biohazard associated with reviving prehistorical amoeba-infecting viruses is negligible, totally negligible, he says. I don't know if I agree with that. Maybe they have a broad host range, right?
There have been, publications, research implicating some of these giant viruses in various human pathologies. None that I'm totally convinced of, but it's not out of the question.
Or maybe they confer a new pathogenesis phenotype on the amoeba.
Yeah. What
Okay.
happens, Dixon, if we wipe out all the latter-day amoeba?
Well, or if the latter day amoebae then can infect us.
Or
Yeah, why
become
not?
pathogens.
I don't see why they couldn't. You can't rule anything out.
Mammoth's revenge.
But anyway, they say people are looking for viruses from mammals in the Vector Laboratory in Novosibirsk, Russia. Remember, Vector Lab.
Right.
Jens Kuhn was talking about that.
And these samples are principally from Russia.
Yeah.
I think maybe all from Russia.
And this
All
is in
for
a BSL-4 facility.
looking at the longitudes.
So they conclude it is likely that ancient permafrost will release unknown viruses on thawing. How long they remain infectious once exposed is unknown, but the risk is bound to increase in the context of global warming.
Now, one thing I was wondering is, what are the odds that these viruses are actually still around and extant, and you just happen to isolate their older relatives?
Sure. Yeah, why not?
Yeah, the other thing that I was trying to figure out that I really couldn't quite wrap my head around was sort of how frequent these viruses seem to be in the samples. So they, I looked at their methods that they're taking 1 gram of sample from wherever, getting this pellet, resuspending it, and then it looks like that pellet from 1 gram of sample went on to 2 petri dishes with amoeba. So what I can't really figure out is how many petri dishes did they have and what fraction of them had a virus in them versus
exactly.
Not having a virus. They talk a lot at the beginning about how common or rare these might be, but you can't quite figure out does every gram of stuff give them something, or did
How
they
many
do
did they
hundreds
have
and
any
hundreds
dishes?
of grams?
Right.
to
Did they
get
have
the
any
9?
dishes that were negative?
Yeah.
Yeah.
It's a good question, yeah. How often, right? All right. So there you go, Jean-Michel. There's your 30 seconds of TWIV. I think it's interesting, though. I do agree, think it's interesting. I like it very much. And I would really... I'd like to see more than amoeba done,
Sure.
right?
There will be. We'll see more.
Yeah.
Paleovirology.
Yeah.
Now,
Good stuff.
I came across this next... it's a snippet. It's a news report. It's a paper. It is a article in Science by, I don't even know who it's by because I've lost
Kai
the
Cooper-Schmidt.
Kai Cooper-Smith. Okay. We made a mistake. It's in quotes. Omicron origin study retracted after widespread criticism. So I thought this is interesting because it shows some aspects of science that maybe we don't often talk about on TWIV. As you know, Omicron is an unusual virus. It had a lot of changes with respect to all the others before it. And so people want to know where it came from. And so apparently a paper was published by Science earlier this month, concluding that this virus slowly arose by gradual mutations, and then we saw it, and it's wrong. It's contamination. They had contamination in their sequence samples, and so they retracted it. But the lead author, Felix Drexler, you may remember, Alan, he was on the TWIV at the BU celebration that you and I were at.
Yes.
For the opening of the needle. He says, we made a mistake and it is bitter. Yeah, mistakes are bitter. But the point here is that apparently if you had looked at the data, like Christian Anderson and Ari, I forget his name, Michael Warby, they said, all right, we could have told you this was wrong just by looking at before you even published it. And that's the point I wanted to make here is that you have computational biologists who say, and why it's wrong is they say, because if this was slowly percolating, it should have accumulated more synonymous mutations, right, that don't lead to changes. And you don't see any of these in the genome. So
the data don't match the conclusions.
Yeah. So
you don't see the right mutation pattern for what they're proposing happened.
Yeah.
And in fact, so this paper came out December 1st and was retracted December 20th by the authors.
All 87 of them.
Yeah, big collaboration. But what triggered them to reevaluate this was the firestorm of commentary mostly on social media and I guess people's science blogs. Because when the paper came out, these folks who looked at the data and knew what they were looking at immediately started commenting, well, no, this doesn't this doesn't line up at all.
Yeah.
And then the question is, how did this get through peer review? And I think there's even a comment in this article that one of the reviewers had raised these very objections, and Science went ahead with it anyway.
Well, I think that's the point I wanted to make, is that it's probably 1 out of 3 reviewers, right? So the editor said, it's 1 out of 3. It doesn't, that's not how it works. It's not a voting thing, right?
Well, and there's a comment from Holden Thorpe, who's editor-in-chief of Science, and defending the decision to publish it. But I think there's going to be some more discussion of that. In fact, I'm sure there's some more discussion of that ongoing in Science.
I mean, you have to pay attention to the minority review. Because I've many times in my career, I had a criticism, and then the editor-in-chief says, well, the other 2 people didn't say that, so I'm going to accept it. So what the hell, right? Why don't you just...
It's not a vote. The question is, did you point out something that was fatal to the conclusion, and if so, it needs to be fixed. Yeah.
So another point to raise here is 87 authors on this paper, and not one of them raised this as an issue. Stop it.
Interesting point,
Yeah.
Stop it.
Yeah, well, they all have a division of labor.
Maybe there were 140 before, and then only the 87 that agreed to publish it.
Well, Dixon, maybe 40 of them were involved in sample collection and not even thinking
was
about.
That obvious?
Right?
Yeah, no, that's true, but that's.
But the computational biologists should have known this, because as soon as it came out, it was immediately picked up. So
well, but it's also, in their defense, it's easy to kind of talk yourself into something like this.
No question.
When you're looking at your own data, you tend to have formed some opinions about it, and
that's true.
You move in a direction. And the point of peer review, in large part, is for other people who weren't directly involved in the project to look at it and say, well, did you think about this? And someone in this article points out that, if this had gone through a preprint, that actually would have helped a lot, because then they would have gotten these comments early on before it went through the whole process.
One of the points that I thought was interesting is, so some of these sequences had mutations typical of BA1, which came after Omicron.
Yes.
The contamination they should have seen that immediately. But anyway,
I do think this is sort of how it's supposed to work in some ways, though. I mean, this isn't a case of retraction because of fraud or something that.
Yeah,
people found an issue and were sort of honest about their retraction. And science as a process is often self-correcting. It has many things that were published that we then realize were not the case. It's just they don't usually happen in 19 days. But now that we have social media, this self-correction has sped up a bit.
Yes,
I agree with
Alan,
that.
you remind me of a Richard Feynman quote. The first principle is that you must not fool yourself, and you are the easiest person to fool.
Yes,
so good. It's so true. But
one of the things I really liked about this is that one of the comments was, if this had been posted as a preprint, it would have been figured out overnight and never would have been published. And I think that's really an interesting. I never thought of preprints in quite that way.
Yeah. Oh, it's happened many times. I remember the paper that came out at the beginning of the pandemic that said, there's HIV sequences in this SARS-CoV-2 genome. Big mistake. Immediately pulled, never saw the light of day.
Right.
So I think a preprint is a good idea. And I also think that the review process needs to address this. But every journal is its own thing, right? They do what they want.
You are right.
Okay, I thought that's a useful little thing to talk about. And now for the main event. By the way, I just wanted to point out, I have a little red light here just in celebration of the holidays. You notice? Right?
Ah, nice.
Little red.
To go with your green chart?
Oh, I didn't even think of that. It was originally green, and I changed it today. I thought I've been doing green the last few days. But yes, you're right. Thank you very much.
It's like when they light up the Chrysler building in different colors.
Well, yes, here in the city, the buildings get different holiday lights. True.
They do that in
Perhaps-
Springfield, too. There's a big office building downtown.
Yes.
Perhaps for this one issue, we should start calling Vincent Rudolph.
Sure.
What do you think?
Call me whatever you want. Just not late
No,
to
you've got
dinner.
a red nose. You've got a red nose right over here.
Our paper for today is an article in Nature. Close relatives of MERS coronavirus in bats use ACE2 as their functional receptors. I would not have used use, but it's not my paper. This comes from
Take advantage of.
what? Well, not take advantage. You don't want viruses doing things. Okay, they could bind ACE2
Yeah, you could.
as their functional receptors. That would be fine. Anyway, this comes from a variety of places in China, Wuhan University, also Seattle, University of Washington, and Beijing. So this is a very interesting paper. So, all right, coronaviruses. Anyone ever heard of coronaviruses? Kind of obscure viruses.
Yeah, don't they like cause some kind of common cold nobody cares
right.
about?
Corona.
All right, I just want to remind everyone.
They infect hops. that they
There are
used to
four.
make beer
There are
with.
four genera: alpha, beta, gamma, and delta. Right? The alpha and beta, the ones that have the genera that have viruses that infect bats and humans. Delta and corona and gamma, mostly birds, sometimes mammals. Okay, so
These are not to be confused with SARS-CoV-2 variants. that are
this
also
is a big
giving
problem,
green
isn't
letter.
it? Yeah.
Yeah.
Do you think they thought of that? No.
No.
All right, so this is about MERS coronavirus, Middle Eastern Respiratory Syndrome coronavirus. So that was the second epidemic coronavirus after SARS-1.
Camel viruses.
Camel viruses, is that what you're calling them?
That's what I would call them.
Okay. This was first discovered in 2012, I believe, in a patient with fatal pneumonia. They isolated the virus, and it was a new coronavirus. And these occasionally infect people from camels, but not everyone has camel exposure. It doesn't transmit very well among people, so it remains, every infection or most infections remain zoonotic, right? They're always coming from some animal, which is good. It has not established itself as a human virus, but it can kill you. It's a severe thing.
Yeah, like a third of the time.
Yeah. So there are just a number of cases every month. I think there have been many thousands since 2012. I forgot the number, but not huge amounts. They're mostly in the Arabian Peninsula, but sometimes they get exported. All right, so MERS corona is a beta coronavirus. And if you've heard Murbeco virus, Murbeco is a subgenus. That's what it is, all right? And they're in camels. It's believed that camels at some time a while ago got this virus from bats, but because there's some relationship there. And so you can find related viruses in bats, especially bats of the Vespertilionidae. I love the Vesper bats, right?
Yeah.
It's a good name. So they're considered to be the source of the ancestor of MERS coronavirus. So this paper is all about receptors. MERS coronavirus binds DPP, dipeptidopeptidase 4, the cell surface enzyme. So ACE2 is a cell surface enzyme. DPP4 is a cell surface, it's a protease. And so is aminopeptidase N, which is a receptor for other coronaviruses. It's kind of interesting. 3 of the 4 receptors are transmembrane proteases.
Yeah, I hadn't really thought about that as a general thing. This sort of gets at a point later that is something that I found really interesting about this paper. And so why, for example, viruses might particularly do evolutionarily well if they bind proteases.
Yeah.
Why, is there some selection for having a protease as a receptor? Wasn't something I thought of until this point.
Yeah. I don't know actually, and this is if you knock out the enzymatic activity of these, whether they still serve as, I think they do. Like you hit the catalytic site, they probably still bind the viruses. Anyway, it's a good question. So some of these bat MERS related coronaviruses, we don't know what the receptors are. They don't bind DPP4. And so that's one of the things they're looking at in this paper. So we're looking at NeoCOV. I think of what's that movie? Matrix.
The Matrix.
Neo. This is the Neo coronavirus. That's the
Oh,
closest. It's a bad virus.
I liked the one that they also talk about, PDF 2180, because I'm pretty sure at some point in time I must have downloaded that.
Yes. I think I have PDF 2180 somewhere on my hard drive.
In your hard drive. So these are bat coronaviruses: Neo coronavirus, PDF 2180, and HKU5. These are bat coronas, and then there's hedgehog coronaviruses, like ERI-CoV HKU31. Neo
Right, so
CoV.
these are all Marbeco viruses
Yeah.
related to MERS-CoV-2.
Right.
They don't use DPP4. So what do they use?
Right, and they're just to hammer it home, they're worth investigating because of the bad viruses out there. They're among the most closely related to the human MERS.
Right.
And Neo-CoV is the closest relative of MERS-CoV, right? So, got that.
That's still pretty far. It was like 85% sequence homology, which is, I mean, that's quite a distance.
So here's the bottom line if you want to go to sleep now.
Oh, these came from bats also.
Yes.
The receptor for Neo-CoV and PDF 2180 is BAT ACE2. And they did some structural work that shows that these viruses bind BAT ACE2 in a different way than SARS-CoV-2 binds ACE2, which is kind of interesting. All right. So what do? You start with sequences of human and animal coronaviruses, and they take some of the spike proteins and make pseudotypes. They take the spike and insert it into a different virus. And ask, can you infect cells with, say, DPP4 or without DPP4? So, MERS coronavirus, HKU4 spike pseudotypes, they can enter DPP4 producing cells. That's what you expect, right? Because that's the receptor for MERS coronavirus.
Actually, there's an interesting sort of sequencing clue early on that I really enjoyed.
Okay.
Because when you do the phylogenetic tree with the whole sequence, they're pretty closely clustered. If you do the phylogenetic tree just with the S1 portion of the spike protein that contains the receptor binding protein, then they are much more distantly related. And if you do this similarity plot that compares the viruses, the biggest difference between the MERS and these guys is in that S1 subunit. So right away, you're suspicious that these guys are using something other than what MERS uses.
Right.
Yeah, I was gonna. It's really striking, and this is an open access paper, by the way. So in the first figure, you go to the sequence part, the A, B, C of that figure, and I think that it really makes a strong case that this spike is something different from the rest of the virus, and they talk about that a little bit later, like how this might have happened.
How that got to be,
So they noticed in their pseudotype experiments that these pseudotype viruses with the spike of Neo-CoV or PDF, they could enter human ACE2 producing cells, which they said was supposed to be the negative control. So this is science, right? Oh, look at this. Make sure it's right.
Yeah.
We didn't do any contamination, but okay, so that's a hint. So then
what's the quote? The most profound exclamation in science is not eureka, but that's funny.
That's funny, yeah.
That's probably the reaction to this.
That's
this
funny.
is something a little different. Whoa! What?
So then they took.
I would have said, "Wait, are you sure you didn't switch samples?"
Yeah, right, exactly
That too,
right.
yeah.
Did you switch it? Repeat it.
Do it again.
Do it
Yeah,
again.
if I were doing this experiment, my response would be, "Oh God, what did I screw up this time?"
Yeah, do it again.
It's always funny when someone says, "This is my result. What should I do?" Do it again, and that's like, well, that's that's not very clever. I say, yeah, but you have to do it.
It's not all about cleverness.
Yeah, do it again.
And Alan, students would say, "Yeah, but do you have something, clever to help me?" No, do it again.
Do it again,
Yeah,
and
that's
then
the
we'll talk
clever
about
thing.
clever.
Do it again.
So many times. I said, yeah, do it again. Okay, so then what do you do next? They have a library of cell lines where each cell line has ACE2 from different species of bats. And they ask which can these pseudotype viruses, NeoCoV spike and PDF spike pseudotype viruses, and then they also compare them to viruses that are known to use DPP4. So NeoCoV spike pseudotype and PDF spike pseudotype viruses entered cells that make ACE2 from most of the Yango-Chiropterian bats. Just say bats. Yango-Chiropteran bats. And very little entry or limited using ACE2 from Yin-Pterochiropteran bats or humans. I think that's what I want.
So these are different suborders of bats: the Yin-Pterochiroptera and the Yang-Chiroptera.
Oh, the yin and yang is. You think
Yes.
they did it on purpose?
Yes, I do.
That's really cool. So, and there's more here. Basically, they're ruling out bat DPP4 as a receptor for NeoCoV or PDF. The PDF virus, just this great PDF.
And they're showing that the ACE2 is a receptor.
Yeah, the bat ACE2, and the human doesn't work very well, but it does work, right?
Right. And so there's something that I didn't quite get here, and I'm sure I'm missing something obvious. From Figure 1E and some other data, it looks like BAT40 is the best, has got the best ACE2 for this virus, right? That's the best entry.
Yeah.
But then they do almost all their subsequent experiments with BAT37 ACE2, which looks significantly worse as a receptor. Am I reading that right?
Yeah. So I mean, they do one of the extended data figures with the bat cells from the BAT40 species. My question was whether maybe, because I'd never heard of that species before, not that I've heard of so many bat species, but that one I really had never heard of, whether the cell lines were harder to get or harder to grow. And so they couldn't do as many in that.
I was thinking it was probably something like that, but it's not. I couldn't find it mentioned, and I was just curious why that was the case.
I don't know. Yeah, it's a good point.
BAT37 was the one that worked with the receptor, and they could work with it.
Yeah,
would make sense.
All right, so then they say the inability of these two viruses, Neo and PDF, to bind DPP4, they say makes sense because the putative receptor binding domain. Remember, the spike proteins of SARS-CoV-2 and all these viruses have a receptor binding domain which binds the receptor. And in these viruses, in the spikes of these two, it's domain B, which they say is a putative RBD because that hasn't been identified as a receptor binding site yet. So they make the putative receptor binding domain, they fuse it with FC proteins, human IgG FC domain, right? And then you can do all kinds of binding assays, including flow cytometric assays, using live cells. There I can use live, Rich, because cells are live.
Yes.
Yep.
And they're not infectious. Although actually in dogs and Tasmanian devils, they can be infectious.
Yes.
And clams too, they can be infectious. And so they can see the binding of these RBDs. They say the Neo and the PDF bound with the highest avidity to pipistrellus ACE2. I had to say that because I love pipistrellus. That is, pipistrellus is Italian for bat, right?
Is it?
Yeah.
You got me.
Yeah. PP.
So the bat's species is bat.
Yeah, bat is pipistrelli is a bat.
That's not the only species like that.
Right.
What is Fledermaus?
Right.
Fledermaus is German, right?
Pipistrelli
You're
is
right.
Italian
The
for
Italian translation of bat is pipistrello.
Yeah.
Pipistrello.
Pipistrello.
And then they can show that
Gorilla
it doesn't bind ACE2, human ACE2 very well. And so based on these, and there's more I'm not going through because you don't need to know all the details. These two domain, the B domain of NeoCoV and PDF2, they say, okay, this is the receptor binding domain, and this is how it interacts with ACE2. With certain bat ACE2, it's really good, and with human ACE2, it's not so good. It has a poor affinity. All right, now they want to do some structural studies. They decide to look at the ACE2 BAT37 ACE2, which Alan was complaining about, not the best one. Well, you know what they say about that? It has a slow off rate. So they thought that would be better for for the structural studies, right? They do say it's a weaker affinity, but the off rate is low, so maybe that would be better. That's why
that was the reason.
Yeah.
Okay.
Yeah, they do say this. I remember that now.
Okay, I missed that.
So they do the structure of NeoCov and PDF RBDs with this BAT37 ACE2. And what they find is very interesting is in that the receptor binding domain of these BAT viruses, Neo and PDF, bind to ACE2 very differently than, say, MERS RBD binds the DPP4. And in fact, the way SARS-CoV-2 would bind ACE2 as well. It's at a different part of the molecule. And if you look at the picture in figure 3C, you could see that the RBDs of here, they're comparing Neo, our receptor binding domain, with MERS, and you see they're binding the receptors in quite different ways. I think in the NeoCov you have these beta strands hitting the tips of the ACE2, and it's the side of the beta strands in the MERS. But the point is that they're different. So they're both binding their cognate receptors differently. So that's the structural information, which I think is really interesting. And the structure gives them the ability to say, okay, these amino acids are important for interaction, so they can then change them and confirm that in fact those amino acids by looking at binding and pseudotype virus entry, and they could pinpoint very specific amino acids that are important for the Neo-RBD to bind the ACE2.
And some of that binding was via glycans.
Correct.
On the receptor as well, right? Rather than direct amino acid contacts, which is also different.
Right.
And I'm just once again reminded of the massive amount of work that goes into a Nature paper these days.
Oh, yeah.
Yeah.
These figures are incredible.
There's a huge quantity of data.
It's crazy, right? Yeah.
And there's another really nice, I'm kind of skipping ahead here, but another nice illustration of what we're talking about in figure 4A, where they show different ACE2 binding spike proteins and compare them to NeoCov, and they show the footprint where it binds on it, and that's a really dramatic demonstration of how different it is compared to the others.
Yeah, I really liked that figure. I had a hard time with the one in figure 3 because comparing MERS and DPP4 versus Neo and ACE2 seemed a little much like too many variables. And here we're seeing ACE2 with different viruses. And as I was explaining to members of my family, it's sort of like one is binding over here and one is binding over here.
No, that's the key here, is that the ACE2 interaction is different. These viruses do it differently from other ACE2 interacting viruses, which is, it's cool, right? Because you often assume that they're going to bind the same way to the receptor, but not always. I'm kind of spoiled by poliovirus because poliovirus and HIV both bind Ig-like proteins in a similar way. And they're totally different viruses. HIV has a glycoprotein that interacts with CD4, and poliovirus is a icosahedral capsid, yet they both hit the same part of their receptors, which are different, but they're both Ig-like proteins. The structure is the same. So, but it doesn't have to be that way.
There's more than one way to skin a virus.
There is. Now, remember, these Neo-PDF RBDs don't bind human ACE2 very well. So they say, okay, let's look at the human ACE2 binding coronas and at the interface and see what's going on with the BAT, with the Neo and PDF. And they could predict what residues might be able to be changed to make it bind better. And they do. And they do some affinity binding assays. So they're just making...
And a reminder that they're working with pseudotyped virus here. So this is not This is not a gain of function experiment that you.
No, it's a pseudotype virus, and also
nuts about.
they're looking at affinity with the proteins, and you can increase.
This is essentially just biochemistry.
Biochemistry.
Yes. Yeah.
Now, if you wanted to do this with the virus, people would yell.
Yes.
And you should have to do that in high containment, and still people would yell because that's the nature of the way
People.
this is going. Yeah.
Yes.
There's some people who just don't want certain experiments done. Anyway, you can change these residues and get these bad RBDs to bind better to human ACE2 than they do naturally, right? More efficient engagement of human ACE2 can increase the binding affinity. They can infect cells with the pseudotype viruses better. So, and they say, maybe these are not the only interactions that are involved. There probably are others as well. And in fact, in the discussion, they say, it's not just about binding, folks. And I'm glad they said that.
I thought, oh, Vincent's going to be pleased. I wonder if he reviewed this.
I didn't, but they must be listening.
Yeah.
To it because they say here, I did highlight it. Everything is yellow, so it's hard to find it when you need it. Right, it's important to find it. Yeah, zoonotic spillover is a complex process involving multiple factors other than receptor recognition. Very good, including spike activation, replication. Hey, replication, that's a pretty big one, right?
That's important.
Yeah.
Immune response, contact opportunity. And so, yes, I agree. It's not all about binding, folks. All right, so back to the data. Oh, then they do some antigenicity. So they want to know, would vaccines protect against these these viruses, NEO, if it ever got into people? One of the things that's interesting is that apparently these spikes are not cleaved. They're not processed upon entry, as are the spikes, say, of SARS-CoV-2, right? It's cut by furins and TMPRSS2 and so forth. But they say we don't see cleavage, at least in the pseudotype, so maybe that it's getting into cells in a different way. But anyway, these pseudotype viruses are not inhibited by antisera from people who are vaccinated with SARS-CoV-2 vaccine or with 10 nanobodies that neutralize MERS coronavirus.
And
Now
that makes sense, since the receptor binding domains are unique for these viruses.
Yes. And that's a good point to make here, is that the vast majority of neutralizing antibodies hit the RBD. There are a couple outside of it, but they're not as good. So it's very interesting. It's all focused on the RBD.
And the antisera they used are from people who were vaccinated with CoronaVac. That's the Sinovac vaccine, which has underperformed, but they do demonstrate in the experiment with the SARS-CoV-2 control that the antibodies from it at least are 100% neutralizing of SARS-CoV-2. So this is completely valid that, those antibodies don't seem to be effective against this virus. But as Brianne said, that's kind of what you would expect based on the structural data.
Now, they do have some broadly neutralizing antibodies that target the stem epitope of the spike, which are apparently blocking fusion, not attachment. And these residues are conserved between MERS, PDF, and NEO, and those antibodies do block entry into cells.
And they mention either here or in the discussion those stem targeting antibodies, since they're hitting conserved regions, would be a good thing to keep on hand.
Yes, we're going to keep them. Yeah. But basically, if this virus spilled over, and it would have to do things to spill over into people, but it's good that we know that they exist, right? So we can keep an eye on them, because the potential is there for them. But as we said before, many things have to be all aligned in order for these to become human pathogens.
Well, and this paper identifies some of the mutations you would want to look for if you isolated these viruses from a human case, and you saw that they had made these changes in their spike, then this is definitely something that might then gain the ability to jump human to human.
Yeah. Now, they make this interesting discussion about how, so the NeoCOV PDF bind a different part of ACE2 than other coronas. So they say this is convergent binding of ACE2 from two different lineages of viruses, and they would say, well, what is the reason for this? What's the function of this? We don't know. What's the evolutionary advantage of ACE2, where you're coming at it from two different places? It must be good for something, and that's an interesting question. They also think
that to me was the most interesting part of this is why has there been this sort of convergent evolution towards ACE2 as a receptor, or going back to the beginning, why are peptidases or proteases interesting receptors?
They also say that this what we have found here supports the idea that MERS coronavirus is a recombinant within the spike gene between a neo-CoV-like virus and a DPP4 using binding virus, which is quite interesting. So, obviously MERS-CoV ended up with the DPP4 binding RBD, but other parts seem to come from a neo-CoV-like virus. We don't know where that recombination occurred. It's quite interesting.
Right, and that aligns with the sequence data where the spike seems to have come from a totally different virus.
Right.
Except that those are actually looking at it backwards because they're comparing it to MERS, and you see that the spike is the different region in these viruses. In fact, what they're suggesting here is that the ACE2 binding spike may be the ancestral spike,
right,
and MERS picked up a DPP4 binding spike somewhere along the line.
So that's the story. I think it's interesting that these viruses are hitting ACE2.
A couple of things interest me. The one thing I have highlighted here is from the discussion, the different interaction modes of the three groups of ACE2 using coronaviruses, along with distinct structures of their RBMs, or receptor binding loops, suggests convergent acquisition of ACE2 used during evolution. So that really interests me. Why would there be, it's as if they're honing in on ACE2 independently, evolutionarily.
Yeah.
As if there's something about ACE2 that's a good target.
Yeah.
And that I can't think. I don't know how to think about that.
Yeah. Is it a sort of biochemistry thing? Is it an expression level? Is it
where it is?
Expression? Yeah.
Yeah. Interesting. The other thing that interests me about this is that the paper comes mostly out of China, including the Wuhan lab,
right?
But is also a collaboration between Chinese and American scientists looking at potential for zoonotic coronavirus infections and digging up ways to think about that, which is exactly what we ought to be doing. This is great stuff.
They also say here, although investigations using infectious viruses will support further evaluation of their risk for humans, the biosafety concern of artificially altering tropism should be taken seriously. Because the antibodies, right, from our COVID vaccines don't work here. It's an interesting addition, right? I think they're trying to head off any criticism.
Yeah,
here.
Exactly.
Good.
Say, yeah.
I hear you.
No, you're frozen.
You're frozen.
Yeah, I can hear you, but you're frozen.
But you probably will unfreeze. That usually is what happens. How long have you been frozen? 30,000 years?
Oh, he's gone. Are you still there, Alan?
He's gone. He's just gone. He's totally gone.
Does that mean his recording is gone too?
Let's start some email and see because he may take a while. Let me take this first one from, all right. Dear TWIV team and Daniel Griffin, we, a group of friends, have made a TWIV Microbe TV donation today in honor of Joanne Jody Schultz, who lived in Boise, Idaho. Our dear friend Lori Schultz, a pediatrician in San Francisco, and twice had her letters read on TWIV, was very close to her sister Jody and held her in great admiration. Jody and hence Lori were instrumental in leading us to TWIV, turning us into big fans. Early on in the pandemic during 2020, Lori was insistent that we all listen to TWIV and that her sister Jody loved it and had been listening to it prior to the pandemic. So Jody is the one who passed here. There's a link to an obituary. She told us so many times that we finally started listening, and we are now loyal listeners too. Lori's sister Jody passed May 27th, 2022 from terminal cancer. And before she passed, Lori wrote in those letters to Twiv. When the letters were read on air, just within days of Jody's passing, Lori was overwhelmed with emotions.
Oh, brother.
Given the impact Twiv has had on all of us and our gratitude from what we have learned, we wanted to make this donation today. Given the role it has played in Lori and Jody's lives, we are making this donation in honor of Jody Schultz. Thank you all at Twiv for your hard work and wonderful insights and for helping us get through this pandemic. So we have Janet Arnesty, MD, Judith Jones, MD, Suzanne Tuckler, and Nancy Tucker, MD. Donations were sent in the names of Judith and Nancy. Thank you so much.
Wow.
Very nice that you're doing that. And very sorry to hear about Jody's passing.
Jody was just 68 years old.
Very young.
It is young.
Dixon, can you take the next one?
I'll do my best. Jane writes, Dear Twiv team, it's a cool 64F/18C and overcast tonight. The clouds are blocking off my view of the night sky in Tallahassee, Florida. I just finished the absolutely delightful episode 962. As an amateur astronomer, the kind with less math, I was cheering along the entire time. Although I spent most of my days thinking about molecular biology and viruses, I still enjoy a good conversation on black holes, and this episode was extra special to me. I was inspired by the episode and picks to give ChatGPT a virus-related task. Here's a poem that ChatGPT generated when I put in the prompt, write a poem about viruses in winter, unquotes. I was impressed with the results and wanted to share it with you. Well, here we go. In winter's cold and bitter air, viruses thrive and multiply, spreading sickness everywhere, a constant threat to you and I. We wrap up warm and shield from cold, but still the viruses find their way into our bodies, young and old, causing misery day by day. Still we hope and still we pray for the end of this dark season when the warmth of spring will come our way and bring a relief and good reason. So let us all be cautious and aware of the dangers that winter brings and do our part to stay healthy and fair, to protect ourselves and those we hold dear. For though the viruses may be strong, we are stronger when we stand together. And when winter is finally gone, we can breathe a sigh of relief forever.
It's pretty good for a computer.
It's very good for a computer. After generating the poem, I went to DALL-E 2, an artificial intelligence made by the same company as ChatGPT, and prompted it to make an image of a virus in space with a galaxy and nebula as a way to celebrate astronomy on Twiv. I'm attaching my two favorite images that it produced. Cheers. I haven't looked at them, but oh yes, here they are. Oh my goodness.
Pretty cool.
I want to add to the space theme right now. My
Can you
grandson.
finish the letter? Can you finish the letter?
I thought I had. Cheers. Oh, that's right. P.S. Okay, P.S. Congratulations to Angela on joining the Twiv team. I look forward to hearing you on future episodes. And she links to ChatGPT and to DALL-E. And her name is Jane, and she's a PhD candidate at Florida State University, Department of Biological Sciences. My grandson, Ivan, who occasionally pops in and says hello, is in a special school in New York City, and they've named each class after some special feature of the class, and their class is named Nebula. Now, he comes home and he tells his parents, my class is named Nebula. And they looked at him like he was from, well, dare I say, outer space. And then they didn't actually understand what it meant. And so, time passed, maybe a half a semester. And I kept thinking about this. And then the Webb Space Telescope issued a picture of the creation of the Pillars of Creation redone, in which you could actually see stars forming. So in Ivan's class, the teachers believe that stars will form, And that is why
very
they
good.
named his class
I like that.
Nebula.
Cool.
And damned
That's cool.
if that isn't a great name for a class.
That's great.
That's
I
great.
Isn't
like
that terrific?
Yeah.
It's good. I think that. I am visiting Tallahassee in April. And I believe Jane is my host. She invited me
wonderful.
anyway.
Wonderful.
So looking
wonderful.
forward to that. These are cool pictures.
Yeah.
Brianne, can you take the next one?
Sure. Judy writes, Dear Vincent, I had to share this story with you. I've been listening to TWIV since March of 2020. I'm a loyal listener of TWIV, Dr. Griffin, and love yours and Amy's Q&A every week. I have shared many TWIV insights with my family. Thanks for making me look so smart in front of my adult children and their partners. They would often ask me for a weekly update during the pandemic. We are fortunate that they come to our house for Sunday suppers each week. It's wonderful to have this time to catch up, and we always talk about current events. Tonight, my son-in-law asked us to be quiet, and he played the beginning of Malcolm Gladwell's podcast. He was so excited for me to hear Malcolm say TWIV was his favorite podcast and that he was wearing a TWIV t-shirt. It was pretty special for me that they were so impressed and excited. Thank you to you and all the team for all you do to teach us and for your dedication and hard work. TWIV is definitely part of the family. And Judy is from Edmonton, Alberta.
Cool.
Neat. I'm going to take the next one because I want Rich to take Phillips. Alex writes, Dear TWIV team, a big thanks again for all the work you put into the various podcasts. They are an exemplar of public science communications. Thank you very much. I thought you might be interested in the following. An ebook entitled Animal and Human Coronaviruses, Evolution and Pathogenicity of the Virus and Disease Prevention has been published. Gives a link for that. This is one of the deliverables from the Euro Citizen EU Cost Action, which is just finishing up 3 years of excellent multinational research into evolution education. The book has been translated into 7 languages and has been awarded a prize as Best Education Resource by the Cas das Ciências Science Education Portal in Portugal. In addition, an accompanying game was developed, also translated into multiple languages. A video about which can be found here provides a link. If you have time and space, it would be wonderful if you could let your viewers know about it. Alex is at the University of Bath. There you go. So done, Alex.
Cool.
Okay, Rich, take the next one.
Philip writes, I was on a Colombian government website helping my daughter enter information regarding COVID exposure and vaccine status this weekend, required for entry. I did this for my wife in May. They asked for travel details and asked the usual COVID stuff. A new question asked about smallpox exposure. I chuckled and thought that was an easy one. I selected English, although I could have done it in Spanish. I did a bit of research. In Spanish, monkeypox is viruela de simio or viruela de monos. Viruela can be any of the poxes. Specifically, smallpox is the plural form, or las viruelas. Merry Christmas and Happy Hanukkah for all the folks at TWIV. And, of course, Happy New Year. So, interesting. The translation into English is ambiguous,
Yeah.
I guess.
They don't want to know
it
about
comes across as smallpox. They wanted to know about monkeypox.
Monkeypox.
Oh, sorry, it's mpox.
Mpox. Yep. All right.
Interesting.
All right, let's do another round. Oh, I took 2. So, Dixon, can you take the next one? Are you there? What happened? Why are you
Yeah,
muted?
everybody's muted.
Yeah. I'm here. Yes, you bet. As I said, muted. Brooke writes, Hi, TWIV team. Longtime listener, and throughout the past couple of years, I've wondered what else the ACE2 receptor is used for, why we have it, and if it's not critical, if there's a way to develop a therapy that binds to the receptor which doesn't change rather than the spike protein which does. Side question, what do the other common coronaviruses attach to? Great question. We answered one of them today. Slide 2 question. Is there any part of the spike protein that doesn't change often that a monoclonal antibody could attach to and prevent the protein from attaching to the ACE2 receptor? close friends. Side question number 3. Does the SARS-CoV-2 virus attach itself to any other receptor? P.S. 12.22C, bright and sunny here in Phoenix, Arizona. As always, thank you for being a resource based on principle and spirit of science. Non-virologists here, but high risk. Captured by your coverage of COVID, staying around for everything else. Always scratches that itch of curiosity, and I am now in awe of the complexity, variety, and depth of these things called viruses. Completely captivated. Thanks, Brooks.
Cool. Brooke got the bug.
Absolutely, and the right questions too. I'd
Yes,
say.
great questions.
So who knows the function of ACE2?
Speak
So
up.
it is angiotensin converting enzyme. It is a protease that is involved in converting a hormone that's involved in things like kidney and blood pressure control and things like that.
All right.
So,
So-
you need this.
Need it, right?
Yeah.
Yeah.
So we can't just say, okay, blow off ACE2.
Right.
That would have side effects.
Yeah, I think that there
Serious.
are some people who actually take medications that also involve ACE2
That's right, that's
for
right.
certain types of cardiac issues.
That is exactly
So
right.
is there any part of the spike that doesn't change often? So the RBD changes often, and that's the part that binds ACE2, right?
Right.
So
Right.
there are antibody binding sites outside of the RBD.
And the
Like
paper we
those
did
stem
today
ones.
used, the paper we
That's
did
right.
today
That's right.
Yeah.
had showed that monoclonal antibodies to the coronavirus, or actually it was to MERS, right?
Yeah, I think it
Spike.
was to the spike stem.
Yeah.
But these,
Still.
I mean, the therapeutically useful monoclonals are all RBD binders
I think that's right. Yeah.
because outside is not as high affinity, the ones outside, unfortunately. They are broader, but I don't think they're therapeutically useful.
Right.
Otherwise, we'd be using them because none of the others are useful right now because of the variants, right? Does SARS-CoV-2 attach to any other receptor? I don't think so. I mean,
Not that we know of.
Not that we know. I mean,
And
there's some
no
reports.
co-receptor that we know of.
There's some reports of a co-receptor, but I just don't know how reliable they are.
Yeah, and I think our paper today mentioned NL63 also binding to ACE2, but I'm not sure about all of the other human coronaviruses.
There are some others that bind ACE2. SARS-1 bound ACE2, right?
Right.
And one of the common cold coronas at least binds ACE2, at least
But
one.
what about the other common colds? Do they bind something besides ACE2 or DPP4?
Yeah,
don't know the
they
answer.
do. I have a chart in my lectures. I will pull it up, and in the meanwhile, Brienne, can you take the next?
Sure. I will stop my Googling.
Yeah, I got it on a lecture here.
All right. James writes, greetings. I just saw your podcast with Paul Offit. Sadly, Novavax was not covered. I was hoping for some information, specifically why it is only authorized as a first booster after the primary series. I am not finding any rationale for this, either with the FDA or CDC. Those that had issues with mRNA vaccine boosters, for those who had issues with mRNA vaccine boosters, this seems like a likely alternative, especially since the bivalent mRNA boosters appear to be underwhelming. Thank you both for those updates and listen every week. I've even mentioned your podcast to a number of folks. Keep up the good work.
So I emailed Paul off and asked him this. And he says they're having another VRBAC meeting on January 26th, and they're going to talk about Novavax. He doesn't
Yeah,
know.
I was thinking Novavax just hasn't asked yet.
Maybe. Paul didn't know. He said, I don't know why it's been delayed, but we're having a meeting and we're going to talk about it. Okay, here are the receptors. So 229E is aminopeptidase N. NL63 is ACE2. OC43 is N-acetyl-9-O-neuraminic acid. And then SARS-CoV is ACE2, MERS is DPP, and of course SARS-CoV-2 is ACE2. And as we learned today, NEO and PDF also ACE2, bad ACE2. It's on my slide in one of my lectures, Brienne.
Very nice.
All right. Rich, did you read one? I don't remember the second
I
round.
read one.
Take the next Jimmy, please.
Jimmy writes, Dear doctors, thank you for your efforts at science education. They're very much appreciated. However, I cringe every time I hear the phrase severe disease. The broader public wants as much protection as possible from disease, even the cases that may classify as non-severe. My poor analogy, if you get a shock every time you use a light switch in the incubator, you would call an electrician. If that electrician started by asking if the shock was lethal, then asked if you were hospitalized for shock, you would find that kind of annoying. Similarly, enough to keep you out of the hospital is not the standard of care I'm looking for. Thanks for your time and attention, Jimmy. Well, Jimmy, I appreciate that. And your analogies are really well put. I think, hospitalization is quantifiable.
Yes.
Okay, which I think is one
That's
of the
right.
major
That's
issues
right.
here. Okay.
Yeah, and everyone counts hospitalization as being the same thing.
Yeah.
If the bigger issue is on the other side of symptoms, is being someone who has a headache or a stuffy nose or a sore throat or being a little tired. Does that count as having disease or not? And I think we all, might call that a little differently.
Right.
So I thought this was useful because, he's right. He's right on.
Yeah.
But as Paul Offit said, we can't prevent mild disease. That's not the way our vaccines are working, especially since the variants can do some evasion and, start replicating for a while before memory kicks in, or T cells or whatever it is, FC-dependent mechanisms. You're going to get a little sick. And the only thing I can say is get Paxlovid as soon as you can, right? Because that gets rid of the symptoms, as Dixon and I know, very quickly. But
That's right.
I also understand not everybody can get Paxlovid, right? They only got it in Brazil last week.
Right.
And many other countries don't have it.
This makes me think of, how much we've learned over the course of this. Because when this started, we didn't know what this was going to be like.
No.
We didn't know that we didn't even know in the beginning that there was going to be asymptomatic transmission. We didn't know what the transmission rate was going to be. We didn't know how it was transmitted. We didn't know how to prevent transmission. We didn't know if a vaccine was going to be possible. We didn't know if immunity was going to be lifelong. We didn't know if it was going to be sterilizing. We didn't know any of this stuff. And now we can have these conversations.
Right.
We didn't
That
know
have all
that there
these
were going
facts
to
in
be.
them.
We didn't know there were going to be variants.
Yeah, exactly.
We said, sorry, coronas don't vary. That's okay.
Right.
Wrong. But unfortunately, Jimmy, I don't think a vaccine is going to do this. I think the antivirals could, and we need more than just Paxlovid so more people can have them. But
Yeah.
I'm sorry, but I do think you are right. And probably a lot of people cringe when they hear severe. They don't want any disease. And
Jimmy, if you get COVID, you can write to us and complain about your disease, and we'll read it and we'll listen.
And we will
it'll
consider
all be
it
good.
severe.
Yes, exactly. However you want it considered.
But the other thing is that hospitalization is also squishy because you can go with or for COVID into a hospital, right?
Yeah. Yeah.
I saw an article where Paul Offit sent it to me, actually, which says that now their hospitals are thinking of not testing everybody for COVID anymore, just testing people who might have COVID, right? So if you come in for a hernia operation, I'm going to test you for COVID because that'll count as a COVID admission, right? So if you're coming in with a respiratory disease and you've got COVID, then you'll be tested for COVID. I think that might make sense. Anyway, one more. I guess no more Alan. Too bad.
Yeah, Alan's gone.
Right. Maybe I should read this one.
You can. Go ahead.
Ted writes, I am struck by the lack of guidance for the old people, those of us over 75. Many of us are spending these days still hidden away, fearful of getting COVID, RSV, and in the dark as to whether or not we will ever see the light of day again. Is there any resource that will tell us by ailment grouping what we can do? What to do when our MD isn't vaccinated and is anti-Paxlovid, and getting into a new PCP is a 6-month wait here in a cold people hand.
No, old.
Oh, I'm sorry, old people land.
Very
How
important.
can we know if our immune system is good? If you are diabetic, what is the prognosis? If you've had a stroke, what is the prognosis? One gets the feeling that over 75 is in danger, but let's not tell them how to evaluate their danger level. So just stay hidden away forever. Not a great life to look forward to. And by the way, I haven't done that. Any resource help would be a public service. I've watched TWIV since March 2020. You have kept me sane. Now I hope you can help me calculate how I can emerge. Many thanks, Ted, Sun City, South Carolina. Well,
Another tough one, right?
I'm 82 years old, and
But your
I just
doctor-
recently got back from Seattle.
Your doctor
didn't stay
gave you
put.
Paxlovid. His doctor won't give him Paxlovid.
Is that well? Then you should get another doctor.
Well, it takes 6
Yeah.
months to get a new doctor. Or start working on it now.
No, well, I think you could get some emergency treatment if you contacted the right group, especially in a hospital.
How can we know if our immune system is good? You can't, right,
Yeah.
Brianne?
You
No,
don't.
you can't. There is no sort of one definition of what a good immune system is like, especially since too much immune system is problematic, just like too little. It's sort of like your immune system is so many things.
If you're 75 years old, your immune system is doing pretty good so far. There's no guarantee into the
point.
future, of course, but at least up to 75. That's
Yeah.
pretty
But
good.
if you're over 75 and you're diabetic, you are formally in a high in a risk category. Okay,
This is true.
And there's not much you can do except for Paxlovid and being vaccinated.
Right.
And I don't know that you necessarily have to lock yourself out completely, but you need to be judicious
Yeah.
about how you interact. And
Yeah, I think
how you behave.
Paxlovid publicity might not have reached down that far to the medical community. That's very sad to see that someone is against it.
It is an interesting question. If you don't have a PCP who's going to prescribe Paxlovid to you, what can you do? And I don't know the answer to that.
Yeah, I mean, he phrases it that his PCP is anti-Paxlovid. So I don't know, the details of that, but
I wonder if you
that
can go
sounds
to
problematic.
I wonder if you can go to an urgent care place and say,
I bet
"Dude,
you might be able to, but I would also raise the issue: would this ever become an across-the-counter drug that you could buy on your own? I don't see the
think so.
downside to that.
think so.
The downside to that.
I think that there are too many contraindications with other medications.
Yeah, yeah. Because it has the ritonavir in there. You can't take it with everything. But I think pharmacies can prescribe it, right?
Yeah, sure.
I mean, if you went,
That's
and
another option. That's
if
right.
you went to a pharmacy and you had a test and you were positive, they could.
like CVS, for
Prescribe it.
instance,
Yeah,
or any of the other
exactly.
large.
But I think
that's a good
Paxlovid
point.
is your way to go because you don't want
That's a
to.
very good
You're not going to get a vaccine every 6 months. Okay, that's just not going to work.
So if you Google urgent care Paxlovid, you come up with all sorts of ways to get
Right.
yourself some Paxlovid
Right.
that don't involve a primary care physician.
Yes, good idea. All right. But I feel the pain. I think there are a lot of people out
Absolutely.
there that are in similar positions who
Yep.
say,
No
wait,
question.
why,
No question.
when can we get back to living?
That's right.
Consider a mask also if you want to get out there, right?
Yeah.
And you're worried about it.
Yes, indeed.
One more from Blue Pilgrim. If we don't do a lot of basic research, then we will never develop warp drives, transporters, tractor beams, synthesizers, subspace radio, tricorders, phasers, or starships like the Enterprise. And the Klingons and Romulans will win all the wars, take over the Earth until the Vulcans get some spare time eventually and decide to rescue us and take over themselves. Someone should tell Congress and the Pentagon. Science fiction generally precedes reality.
That's
I
funny.
love it. I love
That
it.
is funny. That's funny.
Perfect. All right.
That is funny. Oh, I was neglectful.
It's okay. I took care of you, Dixon. Let's do some
All right.
picks. Dixon, you didn't put a pick in. I'm so
That's
sad.
the point. That is the point. That is the point. No. Listen,
the
I've got my
point.
pick right here. I've got it right here. I just have to conjure it up.
You're not at the goat yet, are you?
No, we're nowhere near that.
Okay, good.
Just give me a moment here,
Yeah,
folks.
no worries.
Patience.
We've been waiting for
Take
Alan.
somebody
for-
else. Take
Alan
somebody
must have
I-
anticipated his his failure because he doesn't have a pick listed here.
Okay, right. I've got it. Okay, I've got it. I did the trumpet last week. So today, wait till you see what I paste in there and you'll just be knocked out. Today, we're doing the saxophone.
Cool.
And the alto saxophone. So this is jazz. This is the jazz project. So on an alto saxophone, Paul Desmond, Cannonball Adderley, Charlie Parker. I think Charlie Parker should probably come before all the others. And my cousin, Randall DePommier. Believe me, I'm not putting him in there because of a nepotism or anything. The guy is phenomenal. And if you put him up against any of these others, except Charlie Parker, nobody came close.
Huh.
He was called the Bird for a good reason. He could imitate the fact that he could do the bird songs of any bird species using his saxophone. Well, that wasn't quite true, but Birdland was named after him. He was the quintessential jazz player. Died a tragic death from heroin overdose. Had a rough life, but a wonderful. I think he was born in St. Louis. Fantastic saxophone player. Just an amazing sax player. Paul Desmond played with Dave Brubeck Quartet. Signature album for Paul Desmond alone is the complete RCA Victor recordings, and a signature song with the Dave Brubeck Quartet, of course, was Take 5. Cannonball Adderley, signature album was the Cannonball Adderley Quintet, and there's a song called Mercy, 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 and you will all love it. You felt that way many times when your cultures didn't come out, or they were contaminated, or a lot of other things. The check didn't arrive, you didn't get your grant. Just play this song; you'll resonate with it 100%. And so Charlie Parker played in so many groups that it's impossible to put a signature album on this. Although with Dizzy Gillespie and his All Stars, his signature song is "A Night in Tunisia." Randall DePommier, you can buy one of his CDs. Look him up on the internet. I highly recommend it. And his newest CD, A Midsummer Odyssey, this is his third CD already, and the kid's only 38 years old.
Cool.
Signature song, I Hope It's Spring for You. He has a wonderful touch. He's very, very sensitive. I think you'll appreciate it.
Nice. That's cool. Your
Yeah,
cousin,
so
you said. He's your cousin?
Well, he's probably my nephew. My cousin.
Your nephew. Okay.
My nephew.
I see. There's his picture. Cool.
And he's a wonderful person on top of all that.
You ever see him? Where does he live?
Yeah, no, he lives in New York. I see him quite frequently, and we talk a lot. And he's going to become one of the, I think, mainstream jazz alto sax players of this generation.
All right, Dixon, get him on Twiv and have him play a song.
I will. You got it. Done. That is done.
He can come
No
here to
problem.
the studio, right?
Oh, that would be fantastic. He would love doing that.
You could
He
come
knows
and
about
we can
Twiv. In fact,
have him play.
Guess who his personal physician is?
Daniel Griffin.
Correct. Absolutely correct.
Daniel Griffin is physician to the stars.
He is.
I happen to know one of our listeners happens to be a star, and yes,
Oh, nice. How nice.
who shall not be named because he doesn't want to be outed. But yes, cool. Thank you, Brian. What do you have for us?
I have a story that I had so much fun reading about. It is based on a science paper that came out recently, and the science paper was covered both in the New York Times, which I've linked to, as well as in the Atlantic yesterday. The New York Times article is called "How a See-Through Frog Hides Its Red Blood from Predators," and that one has a really cool video in it, which is why I ended up going with that one. But this is about something called a glass frog, which I did not know about until this here. It lives in Costa Rica.
Oh, it's
and
fantastic.
It is actually partially see-through, is a way to hide from predators. Because a predator will look up and won't really be able to see it, but will in fact see some of its organs, which would look like little blobs, and those little blobs could be anything. They're not sort of obvious frogs. I learned that frogs are quite at risk of predation when they are sleeping. And so this is a particular problem. A sleeping frog that looks like a frog is an easy thing to go after. But if there are just a couple blobs on the leaf, you wouldn't go after that. The problem with being see-through is that you still need to have hemoglobin in your blood. And that cannot be transparent. And so how do you sleep as a transparent frog without having all of your blood obvious and giving you away? And so both of these articles talk about someone observing this in nature and trying to figure out what was going on. And a way that they came up with an imaging strategy where you could actually see this happening. The New York Times actually has videos where you can watch side by side a sleeping frog, a awake and moving exercise frog, and a frog that is anesthetized. And you can see that the blood vessels go away when the frog is sleeping. And they realized that the frog actually has 90% of its red blood cells move into the liver while it is sleeping. And the liver has a reflective coating so that you can't see the liver. And so this keeps the frog invisible, but also it raises another question. Blood shouldn't be able to actually get that close together, all 90% of it in the liver, without clotting, and yet their blood doesn't clot. And so the next question is, how do these frogs not have all of their blood clot, and how do they oxygenate their tissues, both of which could be things that we could get some really interesting insights and strategies from if we could understand them. And so I just found this whole story really cool.
Very cool.
It's amazing.
What's the name of this frog?
The glass frog.
The glass frog.
I learned all about the glass frog, too.
Brianne, the guy who did that work is at the Museum of Natural History in New York City, and he was interviewed today on the BBC. And I just heard that story this morning. It was. I told my wife. I said, "Did you know that there's a frog?" I
was like, who knew about this?
Fantastic, but they can clot their blood when they have to. That was the point he raised.
Yeah, if they have an issue, the blood can clot, but it doesn't clot by that proximity, which in other organisms it would.
So I don't get it. Maybe there's a. You think that's a voluntary action on the part of the frog? Oh my God! I just got malaria. Clot that blood.
Yeah, I mean it's something that's inducible, but
wow,
what it is really an interesting question.
No, it's fantastic. There are a lot of transparent animals out there, by the way.
Yeah, both of these articles are just beautifully written.
Yes, a glass minnow is all. You should check that one out, by the way. It's transparent, totally transparent.
Rich, what do you have for us?
No, we wish politicians were the same way, but they're not. That was a joke, everybody.
All right, Rich, what do you have for us?
So my pick is a book called Mindfulness in Plain English by the Venerable Hanipala Gunaratana. This is my Christmas present and New Year's resolution present to the crowd. This is basically a how-to guide for meditation written by a Buddhist monk. So this is the real deal. I read this book 20 years ago, and my wife and I had a regular meditation practice for almost 20 years. That's fallen off more recently. But I was reminded of this morning because my wife and I were having this conversation about all of the crazy people that we know in our lives and wondering why they were crazy and how crazy they were and et cetera. And it occurred to, I don't want to be condescending in that way, but there are a lot of crazy. Yeah, I'm crazy too, okay? This is the point, okay? Is that it occurred to me that what I learned, a major thing that I learned through this practice is a sort of a new quantum level of critical thinking, and that is not to trust my own mind, okay? I learned that I'm crazy, all right? And I learned how to step back from that crazy and see that it's crazy and not to believe everything that's going on in my head and follow down all of those just treacherous rat holes, okay, when my mind is telling me all of this terrible stuff, is to step back from it and realize, hey, dude, that's all just made up, Okay. And reality is that you're sitting here and you're a blob of, an improbable blob of atoms. Okay. So that my understanding of this all started with this guy, Henepola Gunaratana, who is a very interesting dude all by himself. He is 95 years old. Wow. He's from Sri Lanka, was ordained as a monk at the age of 12. Did missionary work in India before coming to Washington, D.C. at the age of 40, where he ran had a prominent role in a temple in Washington, D.C. And he did that for about 20 years and then started his own meditation and retreat center in West Virginia at the age of about 60. And my wife and I actually went and did a 10-day retreat with him at one point that was of enormous value. At any rate, if you have any interest in this, because I think it's of some value in different ways for different people, and it's, as I say, my justification in a way is that we talk about critical thinking a lot. This is critical thinking in a different sort of context, but critical thinking nevertheless. You're the only one who really knows what's going on inside your head. And it pays to just slow down for a minute, step back, and take a look at what that really is and see what's going on. The link that I've given, you can buy the book. The link that I've given is to an online publication of it. And if nothing else, read the chapter, Meditation, Why Bother?
Yep. I'm reading it right now.
Yeah. And if you find you identify with that chapter, then read on.
I'm reading this and I'm going, holy shit! Look at this. So look at this. It's talking about, you feel cut off, you feel insulated. So what's wrong with you? Are you a freak? No, you're human, and you suffer from the same malady that infects every human being. Chronic tension, lack of compassion for others, including the people closest to you. I think that's the most important thing. Lack of genuine compassion for others. Holy crap. I think that's the basis of every problem in humanity, lack of genuine compassion, right?
That's true.
And it is really in plain, I like it because it really is very much in plain English. And it's written by, this is the original stuff. This is Theravadan Buddhism, okay, as related by somebody who knows. So there you go. Check it out.
Very cool. Thank you.
You're welcome.
All right. My pick actually I got from Cindy last week on immune, and it just blew me away. So I have to share it. It's a paper in the Journal of Experimental Medicine. And it's called Unveiling Skin Macrophage Dynamics Explains Both Tattoo Persistence and Strenuous Removal. Turns out, when you get a tattoo, the myeloid cells in the skin take up the bloody dye and they sit there. And that's why you have a tattoo. And when they die, another cell comes along and eats it and picks up the dye and keeps it there. And this goes capture, release, recapture, and that's why your tattoo stays forever. Macrophages.
Whoa.
Isn't that amazing?
So it's like from an evolutionary point of view, this crap that they've encountered that was injected into your skin, they're sequestering it.
Yes. Yeah.
Right. Okay.
And because it's a pigment,
it's indefinitely by passing it on from generation to generation of cells. Yep. Holy cow.
So that's why a tattoo works. And this paper talks all about it if you'd like to get into the nitty-gritty. But they do some experiments in mice where they ablate these cells and show the importance of them.
So I'm wondering if there's some sort of immunotherapy for removing tattoos.
Yeah, there might be. There very well might be, right?
Or just making your tattoo suddenly go into a different shape.
Yeah, move the cells around. It's very cool. I love it. Science explains life.
You'll have to ask the glass frog how it does it. Because they get tattoos all the time, and this is a piece of cake for them.
We have a couple of listener picks. Rona writes, it was fun listening to your esteemed Canadian guest on episode 946. Wow, her parents raised a pack of amazing children. I'm looking forward to the reveal of the GOAT of jazz, Dr. Vincent. I have a pick for you. I love when people call me Dr. Vincent because they can't pronounce my last name. You need to familiarize yourself with jazz. It's sitting right under your nose. You've never had the pleasure of listening to Columbia's Phil Schapp's Bird Flight? How about you listen to WKCR FM 89.9, Columbia's radio station, the oldest jazz station in America. I know this because I volunteered there during my time at Barnard. Cool, Rona.
All true.
You know about this station, Dixon?
Listen to it all the time. Phil unfortunately passed, but the station lives on.
I guess Bird Flight is a thing, Charlie Parker reference, right?
Exactly right. They played every single song that Charlie Parker ever recorded, and it took weeks. It was amazing. He was very prolific. Jazz is partially meditative. Depends on the level of jazz. If you play this quiet late night jazz, like a piano, bass, and drums, it's very relaxing. I must tell you that it's extremely mind clearing. Let's put it that way. You're so pleased by the endorphins that the jazz induces that you forget your troubles. And Rich, I totally resonate with your position about trying to come to grips with yourself, right? I mean, this latest health issue that I had was one of those times in my life where, this might be it. And now how do you deal with it?
Yeah.
And it is something that every one of us will have to deal with.
Yeah.
And trying to make sense of that. I use an ecological explanation, by the way. If things didn't die, then there wouldn't be new life.
Yeah, of course.
So let's contribute to the new life by dying peacefully.
So I closed my lab so new scientists could have it.
That's right. So, Rich, I hate to trivialize what you were saying, but I had a joke, and I have to tell you this joke. What do Buddhist monks eat for breakfast?
Oh, man. Tell me.
You might know this. They eat omelets.
Jesus.
There's a joke with a Buddhist monk at a hot dog stand in New York. What is it? I'll think of it.
So, Dixon, the jazz thing.
It's very relaxing.
Found myself putting on some jazz and just sitting in a
Good on you.
in a room. Not too long because I'm crazy, but I put on Kind of Blue and some Dave Brubeck. And I realized my laptop has an app called Music. I could get anything because I pay for the bloody thing.
You are right.
And
you are right.
I put Dave Brubeck in, and it plays I could just sit there and listen to this. Nobody in the room, just
Good
me.
Stuff. Turn
I don't
it up
need
as loud as you want.
I'm not bored. I'm just listening to it, and it's feeding my mind. It's really good. So thank you for bringing this up.
You know that there's a you're an Italianophile. I know this because you like things Italian. You like pasta. You like pizza. You like Italian language. There's a guitar player. His name is Bucky Pizzarelli.
Oh, I know Bucky Pizzarelli. My family knew him.
Really?
Yes.
Really? Well, he was a remarkable guitar player, and so you can.
I never met him, but my family knew him. Yeah.
Yeah.
We should talk about him.
I remember the joke. The monk is at a hot dog stand in New York, and he says, "Make me one with everything."
Yeah, that's good. That's very good.
I love it. All
right, we have another pick from Alan. This XKCD just leaped out at me as a great listener pick for TWIV. Proxy variable. Panel 1, we want to study this variable, but it's hard to observe. Panel 2, so we're studying this proxy variable. Panel 3, is it correlated with the other variable? Look, we don't have the funding to answer every little question. Every little question. Right? That's a little question. All right, that'll do it for TWIV 968. Show notes, microbe.tv/TWIV. You can send your questions, comments, picks. You can tell us anything you want. TWIV@microbe.tv. And if you enjoy this program and all our others, please support us. We need your help to continue. Microbe.tv/contribute. Dixon Despommiers at trichinella.org, thelivingriver.org. Thank you, Dixon.
Good night, everybody, and have a wonderful holiday season. It's about to culminate in several wonderful days, so enjoy them all.
It's gone from -1 to -5 in the course of.
By the way, I want
this
you to
show.
know something.
Yes.
And I consider the unit that we've developed here on Fridays as a family. And so I used to not think very highly of making every show. And
I know you didn't.
I came to realize that, this is a connection that you have with other people. You don't just learn about viruses. You learn about outer space, and you learn about mystery novels, and you learn about glass frogs, and you learn about stuff that you never even thought you could possibly hear about. And so I'm absolutely privileged to be among you, because I lack the proper training to actually call myself one of you. But I consider myself a proxied. Alan stepped out of the room, and here I am.
No, you don't have to be at every episode. It's okay.
I'm with you, Dixon. I was anticipating TWIV today, and what was foremost in my mind is that I get to spend a couple hours with my friends.
Yeah, that's the point. That is the point. So that I would never think of this any other way.
I have to say, I didn't imagine that when I started. I just wanted to talk about science, right?
But
it turned out to be more. And I think we are the longest, so we have developed this the most. But I could see the others, getting there. I think Immune is doing pretty well, right?
That's good. That's good
in that sense. But the others too, TWIP,
definitely TWIP.
Brienne Barker's at Drew University, BioProf Barker on Twitter. Thanks, Brienne.
Thanks. It's great to be here. Hopefully you guys are warmer than the 12 degrees we have here.
Hunker down, man. Hunker down.
12 degrees Fahrenheit. Huh? Wow. This went down to -6. It dropped another degree.
Goodness gracious.
Freezy. Rich Conda is an emeritus professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough. Always a good time. Enjoy your Christmas holiday, friends.
Indeed.
I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the American Society for Virology, the American Society for Microbiology for their support of TWIV, Ronald Jenkins for the music, and Jolene for the timestamps. And whatever holiday you celebrate, folks, Kwanzaa, Hanukkah, Christmas, have a great one and be safe. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWIV is viral.